Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
To design drugs that stall the growth of aggressive cancers, it helps to know the structures of the proteins that are revving the cancer's engines. In a series of 3 papers published in Proceedings of the National Academy of Sciences, Scripps Research scientists have illuminated the 3-dimensional structure of phosphoinositide 3 kinase alpha, PI3K alpha, a protein often mutated in cancer cells. We hope that these detailed structural findings lead to the discovery of drugs that affect cancer cells but not healthy cells, says senior author Peter Vogt, Ph.D., a professor in the Department of Molecular Medicine at Scripps Research. That could potentially eliminate the side effects associated with current PI3K alpha drugs. PI3K alpha plays a central role in cell survival and growth. In healthy cells, the protein is flipped on and off as needed. But in numerous types of cancer, including breast, colorectal, endometrial and brain, mutations in PI3K alpha make it active all the time, encouraging the unchecked growth of the tumors. But to do that, you need structural information about what differentiates mutated, overactive PI3K alpha from normal PI3K alpha. This is no easy feat. PI3K alpha is a particularly flexible, wiggly protein, so it's difficult to get a single snapshot of its structure. With this knowledge, they first examine the structure of PI3K alpha attached to the inhibitor. Then, to visualize the protein without the inhibitor, they used cross-linking molecules to attach different parts of PI3K alpha to itself, stabilizing the most flexible parts of the protein. More recently, the research team used the same cryo-EM toolbox to piece together the structure of 2 mutated versions of PI3K alpha often found in cancer cells. That work, published last month in PNAS, showed how some segments of the mutated PI3K alpha resemble the activated form of PI3K alpha. There are quite dramatic structural changes, says Vogt. And in the end, the changes essentially mimic the normal activated form of the protein, with the only difference being that it's always in this active structure. The findings point toward ways to use drugs to shut off this always-on version of PI3K alpha in cancer cells, without turning off healthy PI. His lab group is following up on this research with additional studies revealing how current drugs change the structure of PI3K alpha. In addition to Vogt, authors of the studies, cryo-EM structures of PI3K alpha reveal conformational changes during inhibition and activation. Nanobodies and chemical crosslinks advance the structural and functional analysis of PI3K alpha, and cryo-EM structures of cancer-specific helical and kinase do. Structural studies offer how to guide for designing cancer drugs. Science Daily, the 9th of December 2022. ift.tt/Wacosiefs. Scripps Research Institute. 2022, December 9th. Structural studies offer how to guide for designing cancer drugs. Science Daily. Retrieved December 11, 2022 from ift.tt/Wacosiefs. Scripps Research Institute. Structural studies offer how to guide for designing cancer drugs. Science Daily. ift.tt/Wacosiefs. Access December 11, 2022. Thanks for watching. Please subscribe my channel for more videos.